When a pathogen breaches a cellular barrier, the body has a very narrow window to neutralize the threat before the infection scales. To counter this, pharmacological interventions rely on two distinct clinical strategies. We can either provide the body with an immediate tactical defense, or we can force it to build a long-term biological database. This graph tracks active immunity. Notice the slow initial rise in antibody concentration over time. We introduce an antigen to force the host's own immune system to mount a primary response. The immediate defense is slow, but it leaves behind a permanently elevated baseline of memory cells. If the patient cannot wait for that slow response, we use passive immunity products. The graph now shows an immediate, massive vertical spike in antibody concentration. We bypass the host's adaptive system entirely and deliver exogenous antibodies for short-term neutralization. Because the host didn't make them, that concentration rapidly decays back to zero over time. To source these exogenous antibodies, production historically relied on large host animals. We extract hyperimmune serum from horses that have been repeatedly exposed to a specific toxin, harvesting their circulating antibodies to process for human use. While passive products provide critical emergency neutralization, the bulk of biomanufacturing focuses on the active pathway, specifically the industrial production of bacterial vaccines like BCG and typhoid. The physiological goal of the product dictates everything that follows. Whether the objective is immediate neutralization or long-term memory determines not only the biological mechanism, but the entire industrial pipeline required to manufacture it. The active process begins with the localized delivery of non-pathogenic bacterial antigens directly into host tissue. Local antigen-presenting cells ingest the vaccine and present its antigen to a CD4-positive helper T cell. This triggers a cascade. B cells multiply, diverging into two paths: plasma cells that secrete IgG antibodies for immediate clearance, and dormant memory cells for long-term protection. Because these memory cells now circulate in the bloodstream, any future encounter with the actual virulent pathogen will trigger an accelerated, high-volume secondary immune response. Bioprocessing effectively terminates at the vial, but the pharmacological result is an internal manufacturing event, the generation of dormant memory cells within the host. Replicating that exact cellular response safely for millions of patients requires a highly controlled biomanufacturing pipeline. Step 1 is isolation. Microbiologists select a bacterial strain that is highly immunogenic, meaning it provokes a strong immune response, but strictly non-pathogenic. Step 2 is cultivation. The selected bacterial strain is introduced into nutrient fermenters to multiply. Bacterial growth curves are highly sensitive to their environment. Bioreactor engineers must continuously regulate the pH, internal temperature, and oxygen supply to maintain exponential growth without triggering cell death. Step 3. Inactivation, diverging into two methods. Killed vaccines use exact temperatures or chemicals like formaldehyde to destroy bacteria. Live attenuated vaccines use serial subculturing, repeatedly growing bacteria in suboptimal conditions to genetically weaken them over successive generations. This inactivation phase is the most critical biological threshold in the facility. The process must eliminate all pathogenic risk while perfectly preserving the three-dimensional structure of the surface antigens. If the shape changes, the host's APCs will learn the wrong target. Once inactivated, the batch moves to step 4, purification. Centrifugation and ultrafiltration strip away cellular debris and unwanted biological toxins, leaving only the target antigens. Step 5 is standardization. Technicians titer the purified extract, adjusting the dilution to guarantee a precise, standardized antigen concentration in every single dose. Step 6 is formulation. The bare biological extract is mixed with specific pharmaceutical excipients to ensure the vaccine behaves predictably once injected. The most important excipients are adjuvants, like aluminum hydroxide. Pure antigens often degrade too quickly in the body to trigger a full immune response. Adjuvants bind to the antigen, stabilizing it and irritating local tissue to amplify the APC response. Finally, step 7 involves the aseptic filling of the formulated liquid into sterile glass vials, sealing the finished product from outside contaminants. Through these 7 stages, a raw, volatile biological organism is engineered into a standardized, stable delivery mechanism for human immune training. Even sealed inside the vial, the final product remains thermodynamically fragile. It is a highly sensitive suspension of complex proteins, lipid membranes, and crystalline adjuvant structures. If the ambient temperature rises above 8 degrees Celsius, the kinetic energy inside the vial increases. The delicate chemical bonds holding the antigen together begin to break, and the 3D protein structure rapidly unfolds. This process, known as protein denaturation, permanently alters the physical shape of the antigen. When injected, the unraveled protein fails to bind with the host's APC receptors. The immune system simply will not recognize it. Cold temperatures are equally destructive. If the vial drops below freezing, ice crystals form in the suspension. These expanding crystals physically rupture and shatter the adsorbed aluminum salt structures, separating the antigen from its crucial adjuvant. To prevent both denaturation and freezing, distribution relies on a mandatory logistical infrastructure known as the cold chain. This system of specialized coolers and sensors ensures the product remains strictly between 2 and 8 degrees Celsius from manufacturing floor to the patient's arm. Once this thermal stability is breached, the damage is done. The loss of pharmaceutical potency is irreversible. You cannot refold a denatured protein or reassemble an adjuvant by simply returning the vial to a refrigerator. Manufacturing immunity is a process bound by strict parameters. We must master the microscopic biology the vaccine triggers inside the host and perfectly control the macroscopic physics required to deliver it intact.